Okay, when we talk about T cells, we're talking about cell-mediated immunity. And we're looking at intracellular pathogens. Okay, so when we have a naive T cell, so this is a cell that's just gotten through its little education or whatever, and it's out and about, and it does not see its antigen. It doesn't see extracellular pathogens. Okay, so a T cell always has to be presented its pathogen or antigen, however you want to say. So if we have an antigen presenting cell, it is going to present the antigen on an MHC2 platform. So remember, an antigen presenting cell, so this means a macrophage, a dendritic cell, or a B cell. Okay, so it will present it, and that's how it sees it to bind, and will bind it to the presenting cell. Okay, so once it binds, it has to be activated in some way. So T cells have two flavors, basically. So a naive T cell can either be a CD4 cell or a CD8 cell. So those are the flavors of naive T cells. So a CD4 cell will become a helper T cell. And a CD8 cell becomes a cytotoxic T cell. Helper T cells are very important because we need those to activate other lymphocytes. We need those to activate B cells. So there are more of these, and these are not necessarily more important, but there are more of those, and this is like a key part of the immune system, are the helper T cells. Cytotoxic T cells actually kill infected cells or get rid of infected cells. Okay, so the CD8 cell is going to be activated by a helper T cell. The helper T cell is activated by the presenter, the presenting cell. Okay, so if we have an APC and a presenting cell, and over here we have a CD4 cell. So this is going to be a helper T cell. So it's going to have its little T cell receptor, and over here it's going to present on an MHC2. I guess I'll make it that color here. Okay, so this is an MHC2 because this is an antigen presenter. It's presenting to the CD4 cell, and it binds. the antigen presenter cell actually is able to activate this helper T cell. So activation will occur. Now, the thing that has to be there in order for this to happen is something called a co-stimulatory molecule. Co-stimulatory molecule. So it has to have this co-stimulatory molecule, and if it has that and it's bound, we get activation of a helper T cell. So it is activated. Okay, if we have an APC and a CD8 cell. So the CD8 cell, same thing, is going to have its T cell receptor, and the APC is going to have its platform with its antigen there, and it's going to bind. But what type of platform is this one? This is going to be MHC1, because CD8 cells can only bind to MHC1 platforms on nucleated. These are the platforms that are on all nucleated cells. So it's going to bind on the MHC1. It will also require a co-stimulatory molecule. So the co-stimulatory molecule but that does not for a CD8 cell, that is not enough to activate it. It requires a helper T cell. So I'm going to erase this so I have a little more room here. Okay, so it requires a helper T cell. So the helper T cell okay. And this isn't a CD4 cell. A CD4 cell is naive. This is an activated helper T cell. So here's the helper T cell. Has its T cell receptor, and this helper, or this APC, now can also exhibit the antigen on MHC2. So that one is MHC2. It will connect to the helper T cell. The helper T cell then will send chemicals. So these are cytokines. And again, they're called interleukins. Interleukin maybe 2, I'm not sure. Anyway, interleukins to activate the CD8 cell. Once the CD8 cell becomes an active cytotoxic T cell, then it will go through clonal expansion. Let's come over here. So it will go through clonal expansion, and it will make cytotoxic T cells and memory cells. The memory cells will be able to proliferate if you get exposed again without going through all of this. Okay, this again takes a long time. That process takes a long time. Once you have the memory cells, it doesn't have to go through all that again. You just get the cytotoxic T cells, and then they go and they get rid of the infected cells. Now, the co-stimulatory molecules are extremely important. If they are not there, because there is only a finite number of those, so if there aren't any, then the T cell will not get activated. It will not get activated, and if it's not, let me just put this over here. If it does not get activated, it becomes anergic, and it cannot ever be activated. So once anergic, it can never be activated to be an active T cell. Okay, that's about it. That's T cells. We talked about B cells. And next we'll talk about some hypersensitivity reactions that have to do with B cells.